Good morning. Welcome to today's GRDC Grains Research Update Online. Today's topic is designing sorghum crops for the Australian climate in 2050. And our speaker is Graham Hammer from the University of Queensland.
Thank you very much, Erica. On behalf of GRDC, I'd like to welcome everyone to today's lecture or speech from Graham Hammer. Graham Hammer's from the Poppy at the University of Queensland, and today Graham's going to be talking about designing sorghum crops for Australia in 2050. So a nice blue sky opportunity here to hear from Graham Hammer. So thank you very much, Graham. All yours.
Okay, thanks very much, and good morning to all. I suppose I've been thinking about design of sorghum crops for a long time, and one of the critical things in thinking about that looking forward is clearly that our climate and our experience from history is not going to be the same in the next 30 years. So we've really got to think about the implications of that moving forward. And that's really what I wanted to cover today. And to start off with where we're going to go, I'll just give you the summary of the main messages from the presentation. Firstly, anthropogenic global warming demands that we adapt crops to likely future climate. The future is not going to be the same as the past, and so we have to think a lot about the implications for that in terms of our cropping systems. Clearly, hybrids with high temperature tolerance for seed set are needed urgently, and I think I can demonstrate that in this talk. We have various ways of adapting crops using different types of traits and agronomy, genetics and management in relation to effective use of water, and that remains relevant in terms of improving sorghum crops as we move forward, but it won't be sufficient in terms of where the climate's going, and I'll try to demonstrate that. We also need to be aware that enhanced CO2 is actually a positive for sorghum growth, particularly in relation to its water use efficiency. So there are some benefits from enhanced CO2 that give us a little bit of leeway, and I'll explain that. Unfortunately, that gets counterbalanced by global warming, particularly once we get to about 2 degrees C. And I think we can, from the simulation studies, we can now see that what's going on. So beyond these management of traits and agronomy, I think we really need to start thinking about more novel approaches to improve fundamental water use efficiency. And I think in the future, that's where some of this blue sky research has to go, and where some of our work is already heading. So what I'll cover is something about climate risks and future climate. So I'll talk a bit about what the climate actually looks like and how it's changing. Then I'll look at a crop adaptation study using our sorghum crop model with a number of adaptive traits, focusing on drought and heat risks, go through a simulation study of crop design, which will just look at individual crops, not cropping systems, and then look at the analysis of the trade-offs between yield and production or profit risk in current and future climates. So, and then consider the implications by trying to answer those questions. How do we best adapt traits, genetics, by environment, by management agronomy for future climates, and are there any tipping points? If we look at, firstly, climate risks, there's a few ways to look at that. I think you're all fully aware that climate risk dominates agricultural systems, and seasonal climate variability, particularly where we grow sorghum, is extreme. These are the annual mean rainfall and temperature anomalies for Queensland for the last 100 years. So on the left is the rainfall anomaly. So this is just for the whole state. The average is the horizontal line, and then you've either got positive or negative anomalies from 1910 through to 2018. And you can see they wander up and down. There's wet periods and dry periods through that 100 years. We look at the temperature anomalies from the 100-year average. You can see all of the positive anomalies, or most of them, are in the last 40 or 50 years. And so there's variability going on from year to year, but there are also trends. And one can analyse these patterns by looking at a frequency analysis of these annual data. And so if you look at these frequency components, you can partition precipitation and temperatures. On the left, you've got precipitation, and on the right, you've got temperature. These are the anomalies on the y-axis and the year on the x-axis. And if you take those annual data, you can look at which components of that relate to variability at an interannual level, at a decadal level, which is the green line here, or a long-term trend. And for precipitation, you can see there's a large year-to-year variability, which I'm sure I'm not telling anybody anything about, but that's largely, there's a big ENSO, this is where the ENSO signal, the El Nino Southern Oscillation, fits in, but you've also got a fairly big decadal signal. So there were periods in the 60s and 70s where there were decades that were generally on the wet side. But for rainfall, there's not a lot of trend over that 100-year period. Look at temperature, same sort of interannual variability, large percentage of the variability is interannual. You've got also a big decadal signal, but you've got this massive trend going on. So at the moment in our part of the world, there's not much trend in rainfall, but there's a huge trend in temperature, and you've got these interannual and decadal variability patterns behind it. And the decadal ones are a bit interesting. You see when you have these wet decades, they tend to be cooler. The drier decades tend to be a bit hotter. And so you can, this is a nice way to demonstrate that the trend is actually real, and you can see it once you take out these frequency patterns at different cycles. And if we look at what's going on in terms of global warming, this trend with temperature, which is not just, it's global. Here's the global temperature anomaly. The Queensland one looks just very similar. And what happens here is that this just reduces the relevance of anybody's experience. And it's pretty clear that this is driven by anthropogenic greenhouse gas emissions. So you've got historical, and this picture shows the emissions. So you've got historical emissions up to a few years ago, and then these are all the sort of current policies and pledges of where we're heading. And you can see currently we're heading to a point where we're not really changing, we're levelling off emissions. That's probably about all we're doing. To keep things to a change of 2 degrees or 1.5, we need to get this serious reduction in emissions, and that's where people have been talking about this zero emissions by 2050. And really, that's fairly critical. If we're going to hit 2 degrees, less than 2 degrees global warming change, we've got to get emissions down to net zero by 2050. It's pretty well indisputable. So what does that mean for sorghum crop? We've been spending lots of time and effort building crop models based on the research we've done, understanding the various processes of crop growth and development and the dynamics of those through the crop cycle. So these pictures sort of explain how the crop models work in terms of predicting the water balance of a crop, the infiltration, the evaporation, and its growth rates, photosynthesis, biomass accumulation, leaf area development, partitioning of that biomass into grain, so we can get a prediction of grain yield given a knowledge of the soil and the weather and the management that crop experiences in any one year. And so those sort of models sit inside the APSIM framework. We spend a deal of time trying to make sure that they do sensible things because they are models and they're not perfect things. They give you reasonable predictions. Here's validation of the wheat and sorghum models in APSIM. This is biomass, so this is predicted biomass on the y-axis and observed biomass And so these are experiments where we know we've got high quality data. And the same for yield, for wheat and for sorghum. So we can do fairly reasonable predictions in lots of different situations with different traits, different soils, different environments. So we're fairly confident we can extrapolate into novel environments like those in the future, although we need to take some care about what's in the model in relation to how it responds to high temperatures, for example, where we haven't necessarily got a lot of experience in our data. So what we've done with this is say, well, let's look at sorghum in northeast Australia and do a analysis of adaptation by simulation, because we can run thousands of simulations. You could not possibly do this experimentally. So we can look at historical and future climate. We take 15 sites where we've got historical, long-term historical weather data. We've selected 3 global climate models, and I'll show you those, which ones they are in a moment. And then we run crop simulation, model simulations in AppSim, and we do a range of sowing dates, a range of starting soil waters, a range of agronomy. So we look at generally what would be the productivity across the whole region. We put in the effective water use efficiency from high CO2. So this table over here is a bit small, but if we run simulations into the future, we have to increase this TE, which is transpiration efficiency. So the efficiency with which crop converts water to biomass. By 2050, that's going to be about 20% better than it was in 1990, just because of the increase in CO2. So there's quite a boost to productivity in sorghum just from high CO2. And then we look at a range of adaptive traits. So we've been doing work on heat tolerance, so we can simulate the effect of that. We can change maturity, we can change tillering. So these are genetic things that we can look at alongside the management things to do with sowing dates and density and row spacing and so on. And then we take all that data and analyze it using these yield risk trade-off graphics, which I'll show you. So to look at the global climate models, so here's our locations throughout the sorghum belt, from Quirindi in the south to Clermont in the north, and the main sites that we use for the simulation analysis. We've looked at 33 global climate models, and this is their projections of temperature change in 2050 for October to March in northeast Australia, so during the summer cropping period. And they all project an increase in temperature. The most optimistic one is about 1 degree, the most pessimistic one's about 3. And if we arrange them in order of their predicted increase in temperature, then you can see the rainfall precipitation change is much less consistent, and the ones that have slightly lower temperature forecast have slightly wetter outlook, hotter forecast, slightly drier outlook. So the 3 we chose were a sort of one we call the central in here, which has a next to no change in rainfall, 2 degree change in temperature. An optimistic model which says, well, there's a slight increase in rainfall and about a 1.5 degree change in temperature. The pessimistic one, a slight decrease in rainfall and about a 2.5 degree increase in temperature. So I've used these colours, blue, green and red, as we go through to show the optimistic, central and pessimistic simulations. So we've just used the projections from 3 of these models to represent the range that's out there. If we look at firstly heat stress effects, we've been doing a lot of research on effect of heat on pollen viability and seed set in sorghum, and we now have a fairly good handle on quantifying that. So here's, for example, just a picture to show a control line with full seed set. subjected to a high temperature treatment for a couple of weeks from just after flag leaf to just after flowering. And if you give that temperature sufficiently high, you'll get no seed set. And what we found is there are a range of germplasm, some susceptible. So here's the seed set effect on susceptibles. When your maximum temperature is above about 33, you start to get some loss of seed set. More tolerant ones, it's up to 36. So there's a range of material in the breeding populations across this range currently. This is if you, and that temperature has to happen for about, about a 10 to 12 day period around first flower. And if we put that into the model and say, well, how frequently do we get this maximum temperature or above? And what if we've got susceptible or tolerant types? How frequently do we get yield loss? The way to read this graph is this is the frequency of years over all those simulations where we've got yield loss of more than 10%. And the black one, the black bar, is the historical climate. And 33, 34, 35, 36 are these temperature thresholds where the genotype starts to respond to temperature with seed set loss or sterile pollen. So you can see in the historical one, we've already got 20% of years for these more susceptible types that would have some effect. As you go into these 2050 climates, the optimistic, central and pessimistic, this just becomes over 50%. If you can increase the threshold for that high temperature stress tolerance, you can reduce that effect fairly rapidly until you get up to 35. You've still got some. So if you get up that threshold up to 36, you can just about remove the effect of this high temperature stress on pollen sterility and seed set. And so we're trying to screen germplasm with current Australian Research Council projects, a linkage project with Advanced Seeds actually, where we're looking at germplasm to try and unravel the genetics of this tolerance. The other thing you can do is more about water and look at how you manage the canopy with either tillering, maturity, density or configuration. So some genetic effects or some management agronomy effects. And you can change the canopy development and the water balance by changing all these things. So you can change the number of tillers and you change the amount of leaf area you've got. You can change the maturity. This is a microscopic picture of a apical meristem of sorghum, just as it's changing from producing leaves to producing floral architecture. These bumps on here are the things that become the major branches on the sorghum head. You can change your agronomy with skip row. You can change your density, and all those things change your water balance. So it changes the rate at which you use water through the season and when the stresses occur. And so we then looked at ranges of all those variables and conducted simulation over that entire region and also by sub-regions within the entire region from Central Queensland, Darling Downs, Liverpool Plains. And we looked at these, firstly, we just said, let's just look at genetic effects with a standard agronomy. So if we have 1 metre rows, 50,000 plants, and we've got all these environments. So we've got all these places, the sowing dates, all the soil water conditions, and all the historical years. So it's like running 13,500 experiments with these different levels of tillering and maturity, just to look at what would be the broad effect across the whole region. Now, this is not relevant to an individual farm, it's for the whole production region. Initially using a standard agronomy, but saying, well, what tillering and maturity would give us the best outcome for the whole region? And here's the average yield over those 13,500 simulation experiments. And we plot that against the risk, which is just how frequently does the simulated yield drop below some threshold? And here we've put 1.5 tonnes per hectare as some sort of estimate of a break-even yield. And each of these points is an average of those 13,500 simulations for a different combination of maturity and tillering. The ones that come out on the top here, so they have the highest yield for a reasonable risk, are these early maturity and high tillering. So because we've got lots of combinations of soils and things in here, the overall risk is reduced with this early maturity high tillering type. So get in, use the water quickly. As I said, that's not necessarily the best strategy for every particular combination, but over all of those combinations it is. Because what we wanted to do was look at what happens when you then go into future climates. So you run the same thing with this optimistic 2050 climate, and you can see what happens is you actually do better. Your yield goes up and your risk goes down a bit. The pattern of maturity and tillering doesn't change much. And this is the consequence of that fertilisation from high CO2 that affects the water use efficiency of sorghum. So you get a bit of a benefit. As you go to the central scenario in the green, you've still got a little bit of a reduction in risk because your temperature increase is getting higher, your temperature goes up, the dryness of your air goes up, your vapor pressure deficit goes up, so your water, the effect of your CO2 fertilisation is reduced. When you get to the pessimistic scenario, you start to go over the cliff. So you can no longer, that the benefit you get from that CO2 fertilisation is totally gone, and you get lower yield and higher risk situations predominating. So that's just looking at changing tillering and maturity. What you can do is break that down also, and that was across the whole region. And it looks different if you go into the sub-regions. Clearly, where you've got sub-regions like Darling Downs, eastern part of CQ that are higher yielding, lower risk environments. Generally, the patterns across the climate scenarios are similar. But I wanted to look at one of the more risky environments so we can start looking at this interaction with management as well. So I'll look at just the Darling Downs southwest, which is Gundawindi and Moree. And if we look at this in terms of not just changing the traits, the tillering and maturity, but say, well, what if we change population, row configuration? So each of these points is now, for this region, all of those, each point has a different level of tillering, maturity, population, and row spacing. And the line here is the original one from the initial simulation, and that point was the one with the early maturity and low tillering, the optimal one, if we didn't change management. If you go into this region and you can also change management, you can see you can move this way. You can keep the same yield level and reduce your risk by reducing your population, reducing your tillering, and going to single skip. And in future climate scenarios, your yield goes up a bit here, but it comes back down here, and again you go down once you get to this pessimistic climate future. But all of these things in this environment tend to favour less intensive management, reduced population, reduced tillering, and some single skip systems. So you can adapt those optimal types by changing some genetics and management locally, and that would give, and there would be different answers in different regions. So you can improve your position by changing this genetics by management. I'm probably not telling anybody anything, they're already doing a lot of that. But if we look at what happens into the future, that's still the case, but you can't overcome this the change of the climate scenario. So these things are useful, but they're not sufficient to overcome what's about to happen with climate change. So what does that mean? Just to summarise, we know heat tolerance is important, but it's going to become more important, and breeding solution is really the most robust way to overcome that, although that needs to be considered in the cropping systems context. And I know there's work going on that many of you are probably aware of with the early sowing, and I think that's another opportunity for the heat tolerance avoidance, but I think we're going to need both of these things. The climate scenarios generally shift these frontiers, but they don't, you can't change your genetics and management with what we've got currently to overcome the climates that we're going to experience by 2050. And that's because of this balance of these effects. The CO2 fertilisation effect, in fact, gives us a bit of a breather, but we're going to run out of it soon in terms of being able to adapt to future climates. So the existing concepts are relevant, and so this idea of optimising G by M is a good one, but we're going to need something beyond that because it's just going to get harder to increase productivity. And so you know, especially if we get to this greater than 2 degrees C climate, global warming, that's often mentioned as a tipping point for lots of things. It seems it sort of is a pretty relevant number for what might happen in terms of tipping sorghum into productivity and risk issues as well. So we've been looking at enhancing transpiration efficiency as one of the few traits that's showing promise. And if we do the simulations by changing both the light use efficiency with which sorghum plant converts light to sugars, or the transpiration efficiency, biomass per unit of water, on these yield risk trade-offs, and we just look at the historical climate one. It's a bit hard to see. This is the current situation where you've got existing light radiation and water use efficiencies. We increase the radiation use efficiency, we actually don't get a huge effect. In fact, risk goes up a bit. If we increase the water use efficiency, if we go down this way, TE goes up a bit, we can significantly increase the yield and reduce the risk. The interesting thing when we move to the 2050 climate scenario is that is somewhat maintained. So this issue of improving water use efficiency is probably one of the few things we've got that can give us yield advance into anticipated climates. So we've had a research focus on that now for a while, trying to understand the physiology and genetics of variation in transpiration efficiency by doing silly things like growing sorghum plants in hospital pots so we can measure exactly how much water they use and when, and doing detailed measurements on their photosynthesis and conductance, and doing these studies across David Jordan's pre-breeding population so we can not only understand the physiology, we can understand the genetics. So just to reiterate the take-home messages, you know, we really do have to think about the consequences of anthropogenic global warming, particularly the high temperature tolerance aspects. That's already, you know, it's needed now and certainly well before 2050. The concepts of adapting traits and agronomy remain relevant, and it's important to optimise those systems, but that's not going to be enough by the time we get to 2050, especially if we're up in the 2 degree range. Enhanced CO2 gives us some positive effects for a while, so you know, it might not look as bad as it could be, but it's going to hit, particularly once global warming gets to 2 degrees. And we think these novel approaches to improving fundamental water use efficiency are a good option for researching. So thank you for your attention.
Graham, excellent presentation. My name's John Cameron. I'll be facilitating the Q&A session for this afternoon. So this morning, I would encourage anyone out there to get your questions in using the question tab on the event software. Professor Graham Hammer, what a great presentation. There's some challenges ahead, definitely. We've been hearing a fair bit of late about early sowing of sorghum as a strategy to avoid the heat stresses at flowering and pollen sterility. So how does the work you're doing marry with that or relate to strategies for early sowing? Does it relate to it?
Look, I think they're probably complementary. In a sense, the sort of global warming issue will enable the sowing window to open up. And even without that, I think the work that's going on looking at trying to sow early into colder soils will give you a flowering time that will give you less risk of heat tolerance, of heat stress. So I think it's a sensible strategy. We're going to need all the irons in the fire for dealing with this. The issue, of course, is you never quite know when you're going to be able to sow, and so you can't always guarantee sowing early. There will still be lots of situations where one's sowing in September or later, and so you're going to be subjected to these heat stress events. So I don't think it's an either or. It's really these are all things that are worth pursuing.
Graham, in the modelling work that you've done looking at these future climate options and the response of the plant, you've definitely been looking at the factors of pollen sterility and heat stress and how those varieties respond to those increased pressures. Have you also been accounting for the higher variability that we're likely to see in future climates in terms of more storm rainfall, fewer rain days, and the impact that might have on water storage in the system?
Yeah, look, the water balance takes all that into account. So when you get the scenarios from these global climate models, at the moment, they don't give you detail down to that level. You sort of get what's the sort of monthly shift in the amount of rainfall you're going to get. And as you can see from what we saw from those models, the rainfall effects are not that huge. The predicted rainfall effects for the sorghum region are not that great. The major effect is actually not through how much water is coming in, but how much is going out because of the increase in the temperature driving higher vapor pressure deficits and higher evaporation rates. So you sort of it's more driven by demand, the amount of water use rather than the amount of water input. So at the moment, probably the answer is, look, we take into account what we can from the models. They give you shifts at a monthly level rather than changes in storm intensity and things like that. So, we haven't really input in here changes that would be on intensity or something like that. The rainfall amounts would just be moderated by the overall average increase predicted or decrease predicted for a particular month.
So in that same vein, there's a question from Glenn out near Dolby. G'day, Glenn. Can starting with more moisture in the soil profile and using a higher trigger for when you make your decision to sow than we used to use, can those sort of strategies compensate for these higher temperatures and changed environment risks that we're seeing in the future?
Yeah, it's a good question because we haven't clearly. If you use a higher trigger, you're putting yourself into a lower risk situation. and so that may well be a sensible strategy. As I said on the way through this, the analysis I put up there are averages over a large number of sites and soils. So really, the answers and the sort of optimal combinations will be very different for different locations and soils. But certainly, I think the lowest soil trigger that we used in those simulations was, I think, 75 or 80 millimetres. So there was, there had to be a certain amount of stored soil water in there. So I think that's a good point. If your demand is going up, you at a sort of a farm scale, field scale, clearly, if you increase your trigger point, you're going to put yourself into a different yield risk situation, and that would be a pretty sensible way to look at it. And one could simulate those situations for specific cases.
There's a question that's coming from Daniel at Toowoomba. G'day, Daniel. He has a bit of a vested interest in cold tolerant sorghum, and the question he's got, well, what about breeding for cold tolerance in sorghum? And so we can say the crop earlier, how much of a response do you think that could be there, and how easy would it be to breed for those sort of traits alongside some of the other abiotic stress tolerances you're breeding for? Or has it become problematic to put too much into your breeding objectives?
Well, the more you put in there, the harder it gets. But I think that's a sensible question. So to go to the early planting and to encourage or to open up that window, which makes sense. There's variability in that cold tolerance, and so it's certainly something that could be bred for. Some of our sort of inclinations are that the high temperature tolerance and the cold temperature tolerance, certainly for seed set, are a bit linked. I'm not sure that there's any connection with the seedling growth, but certainly it's a potential breeding target, and one that one could phenotype for. I mean, certainly with things like maize, for example, it's much more cold tolerant at those early stages. So it's reasonable. It's a reasonable breeding target.
Okay, well, staying with the breeding targets, Graham, g'day, Graham, sitting probably on a beach up in southern Queensland somewhere, has come in with the question of how complex is the genetics of polar and heat tolerance and improved transpiration efficiency? So, how difficult do you think it's going to be to identify these traits and get them into hybrids that farmers can use? What's the science risk and opportunity?
Yeah, okay. It's tricky. These are not, they're not single gene traits. So these are not magic bullets. But we know with the heat tolerance, we're probably not at a point to have the genetic architecture fully worked out, but we're a fair way into phenotyping populations and starting to do those genetic analyses. So we'll be able to see how complex the genetic architecture underneath it is. But with modern techniques of genomic selection, even with complex traits, there's good potential for improvement if we can reliably measure germplasm sets that are a good training set for the breeding populations. So even something like transpiration efficiency, what we're trying to do is find ways that one can get indexes that measure its variability across lines fairly quickly, and that way you can then start to link it in with modern approaches to trait improvement in breeding. So they're complex traits is the short answer. We'll probably never really fully understand the mechanisms and the genetics and physiology of all of it, but you can still breed for it if you've got a good handle on the genetic architecture underpinning it, and I think we can sort of move in that direction.
So you mentioned there the transpiration efficiency methodologies to better understand and compare varieties for transpiration efficiency. The other thing you were looking at was improving radiation use efficiency. Now, you've been cited as saying that the old models of genetics by environment by management are not going to cut it. If we dig around at the cellular, you're wanting to actually understand some of the cellular functions at a better level, how the plant architecture works in the roots and the leaves, how photosynthesis works. Can you unpack that a little bit for me in terms of where you think there might be some gold leads to follow?
Okay, yeah, look, we've been doing some fairly detailed work on this as part of the ARC Centre of Excellence studies, and using Sorghum as one of our models. And I think what it's sort of showing is the sort of conventional view of trying to improve photosynthesis by making it more light use efficient is probably not the highest priority target, unless it's connected with enhancing water use efficiency. So we really need to connect on the components of the aspects of photosynthesis that are also relevant to improving water use efficiency. So the actual conductance and the regulation of stomata, and how you can try to get an enhanced photosynthetic rate without changing your conductance. And it's possible there are parts of the pathways that can be manipulated that enable you to do that, but they're not necessarily the parts that are associated with the actual capture of photons of light, if that makes any sense. So there are opportunities in there that I think are coming out of some of the more fundamental work now, and we'll certainly have been looking at trying to measure these things on large numbers of entries in the field, so we can again try to link it back to genetic architecture. So this, looking again with David Jordan's populations in field trials, trying to assess where we've got natural variation for some of these aspects of photosynthesis and water use efficiency.
Question from Laurie at Toowoomba. G'day, Laurie. Genetic traits to enhance temperature tolerance, high temperature tolerance during flowering in particular. So are there specific traits that you're looking at here, or is it more a case of just exposing lots of genetics to high temperature at that time and seeing which ones pops out, or are there specific traits that you're targeting in the research?
Yep, certainly we're trying to identify what underpins the effect. So one can do that in a couple of ways. So we certainly have to control the exposure of plants to particular high temperature challenges at specific developmental stages. So we initiate the effect on mostly pollen sterility, but it appears it's not all pollen. Some of it can be on the female parts as well. So we're looking at trying to understand how much is pollen, how much is the effect on the female parts of the flowers, and then there are things that can be measured that give us an indication of that. So at this stage, we're just exposing lots of different lines to challenges, looking at the variability and trying to determine what is it that causes the variation in those responses. And if we can come up with something on that, then we would have a quicker way to measure it across lots of other material. So at the moment, there isn't something. There's there are a number of thoughts about what might be causing these effects on pollen sterility, to do with effects of temperature on membranes and various proteins, but none of those have been really, we haven't got to a point where we've got an assay of some sort.
Yeah. A question for Michael, and it relates to: have you got enough toys to play with? So if you look at the sorghum, the sorghum genome and how much variability is out there in the wild types and the various commercial varieties that are out there, do we have enough genetic variation to select for the traits that you need, or do you think we might need to go to gene editing or some of the more GMO type techniques in future?
Yeah, look, I mean, one thing about sorghum is a remarkable variation in natural variation, native variation, and the very nice thing about what the crew at Hermitage have done with David's program is really introduce an amazingly diverse range of material in sorghum from parents from all the major races across the planet. So we have an amazingly diverse germplasm set to play with, and the linkage with that program is really enabled us to do our work remarkably differently in terms of understanding the genetics. If we do get to a point of understanding it, then clearly there may be opportunities for things like gene editing. But I think in these more complex traits, that's unlikely. You're more likely to be able to move into more quantitative approaches using genomic selection, and with the prediction of what might happen with different sets of genetics from developing models based on understanding the responses across all of that variation. So I think the variation is there. We've got the germplasm. What we haven't really got are the ways to assess it quickly. So that's probably the gap.
So I think you might have already answered this one, but it's from Scott, who says, great presentation. Thanks for that. But do you think that identification and implementation of molecular processes that improve responses to temperature, how big a role will they play compared with the modification of phenology or crop management? And based on your modelling, do you think we need to look beyond the responses or the genetic variation that might already be in sorghum? That's sort of, I think you've already answered that, but any additional comments there? Or?
Yeah, I think the variation is there. It's obviously probably not in the sorts of material that anybody would want to grow to produce a profitable sorghum crop, but it's there and can be manipulated. So I think the phenology crop management component of that, I think we've covered that a little bit. There may be opportunities to extend the sowing windows so you change your exposure risks. But when we've looked at the analysis across times, these heat events can happen fairly common across just about almost any time that you would have sorghum flowering, except from exceptionally early sowings. And I think the genetic variation, I said that I'm inclined to think that with the material we've got, we know that there's a good deal of variation in it. And there's probably enough there to play with to work to get some sort of practical solutions to this with what's available without going to gene editing. But having said that, if we knew what the mechanisms were and it was pretty simple, you might look at gene editing, but my feeling is it's unlikely to be simple.
Okay. There's a question from Glenn at Dolby again. So with breeding of varieties and getting the genetics right for what we're trying to do, or the challenges we'll be faced with in the future, and indeed are being faced with now, there's a bit of a gap in the lead time between the pre-breeding research and when those new traits are reflected in locally adapted cultivars. Sometimes that can be 10 or more years. So what sort of breeding can you tap into to get this started now, and what are your comments on the breeding program and the lag time to see this research in current varieties?
Yeah, look, it's a good question. Certainly the 10 years is realistic, and this is why, you know, thinking about climates in 2050 is not absurd. You know, certainly any of this material, any of these sort of enhancements are unlikely to be in anybody's field much before 2030. So it's you know, it's pretty important that we get it right. And the how does one say this? The linkage between the physiology work we've been doing and the development of the pre-breeding germplasm pools at Hermitage has been critical in being able to do this. That's probably not going to happen into the future.
Okay. Miranda has come up with an excellent question here, complementing the discussion on radiation use efficiency and transpiration efficiency, and unpacking that. What about responses for water use that we can do with agronomy? And you've mentioned a couple. Row width modifications was one that came up occasionally. Often that's associated, if you have a better year, the wide skip row configurations often lose a bit of yield potential, but perhaps can even it out in those low yielding years. But then we're looking at lower tillering varieties, and the other trait you mentioned was lower population densities. What you want to comment more on the agronomy tools and levers that we've got?
Yeah, look, I probably went through all that fairly quickly, but all of those options were included in that adaptation study, and they all have an effect. And so that was the point about look, the G by M optimizing your traits by agronomy is still important for a specific situation. So, and the point someone made about, you know, trigger points for how much water you've got to have before you sow. So knowing your soil water immediately changes your risk profile for various combinations of those factors. So that sort of localized tweaking is critical at that local level. In terms of just getting the risk return trade-off right. But overall, what I was trying to get at is we sort of understand that. We know what that can or can't do. And when we get to that sort of 2050, if we've got +2 degrees, none of that is going to enable us to get back to where we are now. So you can't moderate those factors and change enough of them to overcome the issue of the global warming. It gets up to 2 degrees.
Yeah. Question from Solomon. Do you think there's any synergy between heat stress tolerance and radiation use efficiency? Interesting question. Are they linked?
I can't see any reason why they would be, but the proper answer is I don't know. But I would think it would be unlikely because they're remarkably different processes, one being developmental in the florets and the other one being more a growth process in the leaves. So I can't see why there should be any connection.
And we might make this the last question. because and I know there's a few that we won't get to ask and answer, but I think most of them have already been covered by responses to other questions, but we're just going to run out of time. The last one relates to science risk, and you're looking at some basic plant physiological processes all the way down to transpiration efficiency and how photosynthesis is working within the plant. To what extent do you see that research as blue sky opportunity, or do many of those paths already have a bit of a roadmap as to a lead where the gold nuggets might be found? If you want to just comment on the risk and the rewards of this type of research.
Sure. I think this is where our connection with the Centre of Excellence, ARC Centre of Excellence in photosynthesis has been pretty useful, in that it's enabled us to think more about the fundamental processes and the implications of modifying them and actually developing models to do that. And so in some ways, that's given us a bit of a view of what we think is a sensible way forward and why we're pushing on this, the water use efficiency aspects rather than the radiation use efficiency aspects. So that's, I think it's still high risk because we don't really know how those things work all that well. But we know there's variability for them. And so we know we can probably do something with it, even if we don't fully understand it. But one can push the more fundamental research into that space. I think that makes good sense. But to me, that's in terms of future climates, that's where the big returns can potentially be. But they're certainly harder to get. But we can sort of, rather than just go at it in a random way, I think by connecting understanding at the crop scale back to the biochemical scale, we can sort of see where are the more sensible routes to take. And that's one of the things that I think's come out of that Centre of Excellence work.
Graham,
So
fantastic presentation. That does answer my question. We've had a number of comments that are coming through already just saying what a great presentation, how much they've enjoyed it. Great to have a look on a topic that's way over the horizon. It's out there and pushing the boundaries in response to the challenge, so we can see what we've got to do to keep our industry sustainable under the challenges posed by future climates. Graham, thank you very much. We're going to close the webinar off now. Before we do, I'll hand to Richard Holznick from the GRDC for a quick wrap up. And just a reminder that as you're exiting the survey, a pop-up will appear with a quick exit survey. We really value your feedback, and if you could take a moment, it'd be greatly appreciated. Richard Holznick, over to you.
Excellent, thank you very much. Firstly, I'd like to really thank Graham for his great presentation today. I think it's great to see some of those future horizon topics that are being discussed, especially designing sorghum for 2050. It shows that we do look into the future, and it is a good example of something that's out there in sort of blue scope, but still trying to achieve it by minimising our risk by associating those traits that we're looking for with water use efficiency to make sure we're maximising productivity for of our sorghum in the future. So great, thank you to Graham Hammer from the University of Queensland, POFI. Thanks again. I'd also like to thank John Cameron and also Erica Mackay for their great facilitation and also organisation of the webinar series. And lastly, I'd like to welcome everyone to Thursday's webinar, where we're going to be talking around future farming. So another futuristic type topic, but we're actually going to look at the technology base this time. Our first speaker will be Derek Long from the University of Southern Queensland, and he's going to be talking around pest detecting and scouting and use of machine vision apps, with a case study around cotton, but also what the implications are for grain. We'll then also hear from Alison McCarthy from University of Southern Queensland, and she's going to be talking around machine vision and the application of those to nitrogen management. So with those two topics, we move to our tools and technology section. But thank you very much again for today, and hope you join us next week. Okay, catch you. Bye.